still missing some attendees, but we're going to go ahead for the sake of time, begin our webinar. Again, if you have any technical concerns, please feel free to private message me at the Global Genes host. You can also email me at Ashley Y, ASHLEY, at globalgenes.org. So again, if you are joining us late into our webinar, it is being recorded and will be provided to Softbones. And so you can go ahead and contact Denise Goodbar if you have questions regarding the recording. So I'm going to go ahead and pass this off to Deb to get us started.
Great. Thank you, Ashley. Welcome, everybody, and thank you for joining us this afternoon. I'm Deborah Sucheg, and I'm the president and founder of Softbones. And as many of you know, my son Cannon, who's now 10, has hypophosphatase, and I'm also a carrier of HPP as well as a caregiver. Many of you know that education is a key part of our mission at Softbones, and this webinar is just one way that we can reach patients around the world to educate them not only about hypophosphatase, but the role that alkaline phosphatase, the enzyme, plays in our disease. Next slide. And today's webinar would not be possible if it weren't for Ashley, who you just heard from, and our partnership with Global Genes. For those of you that don't know, Global Genes is one of the leading rare disease patient advocacy organizations in the world. And the nonprofit organization promotes the needs of rare diseases in a unifying symbol of hope with their blue denim jeans ribbon, as you can see here. And it began as a grassroots movement back in 2009, around the same time that we started Softbones, with just a few rare disease patient advocates and foundations, and it's grown to over 500 global organizations. So we're able to bring this webinar to you through the use of their technology and their support. So we want to make sure that we acknowledge them and say thank you for being a trusted partner. With no further ado, the star of today's show is no stranger to many of us. Dr. Michael Whyte is a professor of medicine, pediatrics, and genetics at the Washington University School of Medicine, and he's a medical staff member of Barnes-Jewish Hospital in St. Louis. He's also the medical scientific director of the Center for Metabolic Bone Disease and Molecular Research at the Shriners Hospital for Children in St. Louis. And of course, in his spare time, or what there is of it, he serves as the chairman of our Softbones Scientific Advisory Board. Dr. Whyte has authored hundreds of scientific papers, book chapters concerning pediatric and adult metabolic bone diseases. He's involved in the research for some of the potential therapies to treat our diseases. And at the end of his 20-minute presentation today, we've allotted 10 minutes for questions. So Dr. Whyte can't answer any personal questions or offer medical advice. So we ask that you please keep your questions relevant to the subject of ALP within the context of this presentation. And with that, Dr. Whyte, thank you for joining us today, and we hand it over to you to educate us on the ABCs of ALP.
So to all attendees, if you have questions, we can go ahead and utilize the Q&A portion at the bottom of the screen. It's on the right-hand corner. I will go ahead and filter those over to Dr. Whyte at the end of his presentation. So I will go ahead and switch this over to Dr. Whyte.
Right. As I was saying, I'm grateful to Deborah Sitzik and Denise Goodbar and to Global Genes for making this webinar possible. I've been asked to talk about alkaline phosphatase, the enzyme that's deficient in hypophosphatase, using lay language. So this is to help especially patients and their families understand what alkaline phosphatase is and does. And I alert you to, let's see, I'm trying to change this slide, that just this month, April, Jose Luis Millan and I, Jose Luis at the Sanford Burnham Institute in San Diego, and I published this review in Calcified Tissue International of alkaline phosphatase and hypophosphatase. This is written in scientific language, so if you want to share it with a physician or a scientist, by all means, it should be readily accessible to all. So an up-to-date review of what we're talking about this afternoon. My university, Washington University School of Medicine in St. Louis, tells me to disclose to you that I have honoraria. I get travel reimbursement and have research grant support from Alexion Pharmaceuticals. They're in Cheshire, Connecticut. And many of you know that back in 2015, last year, aspartase alpha, which is also called Strinsic, was approved in the United States, the European Union, Canada, Japan, as a biologic for treating pediatric onset hypophosphatasia. But the story of alkaline phosphatase really begins at the start of the last century. This chemist, Robert Robeson, had finished some work during World War I, and then at the Lister Institute in London, England, turned to what was a hot topic of that era, and that was how do we use carbohydrates, sugars, other chemicals to have energy for metabolism, for our bodies to function properly. And in that era, the chemists knew about what's on the next slide, and that is phosphate. This is a phosphorus molecule hooked to oxygen and hydrogen. A formal name for it could be inorganic phosphate, abbreviated PI. But the chemists knew about phosphate back then, and they also knew that phosphate could be coupled to sugars. Robeson was interested to know how the sugars might be released to be free for energy metabolism, getting rid of the phosphate here, and studied growing rats and rabbits, looking for some factor that released the phosphate from the sugars. And when he looked in the bones and liver of these rats and rabbits, that's where this factor seemed to be especially rich. In the test tube, the phosphate would be released from the sugar, and the sugar would be then free for energy metabolism. But he was a wonderful chemist, and he realized that the release of phosphate might have significance beyond energy metabolism and be relevant to bone mineralization. He knew here that bone is like a living fiberglass. We think sometimes of bone having a fiber. It's a protein called collagen, and that in good health there's deposition of the glass, if you will, and this is crystals of calcium and phosphate combined. And Robeson and the chemists of that era knew that the amounts of both calcium and phosphate had to be just right to have a strong, resilient skeleton. And then in 1923, Robeson published this paper, and here he speculates, and I'll translate it from sort of the medical language. He's talking about the possible significance. So maybe sugar hooked to phosphate was important in ossification, in the formation of bones, thinking that sugars, with this factor that took the phosphate off the sugar, was releasing what? Phosphate that was necessary to combine with calcium and therefore maybe put it into bones. So he sort of went from, or continued an interest in energy metabolism, but now was onto the notion that there was something in bone and liver that could promote mineralization of the skeleton. And you could consider this or call this Robeson's enzyme. But what is alkaline phosphatase? So we like to abbreviate it as ALP. I say that this is an enzyme, so you say, Dr. Whyte, what is an enzyme? You keep saying that, but tell us more about what enzymes are and do. And I would say to you, an enzyme is a protein that does what? That breaks down certain sort of target chemicals. So it's a catalyst in a way. It's a protein. Its job is to break down certain specific chemicals. It depends on which enzyme you're dealing with as to which chemicals are going to be broken down. But these are then called substrates. So they're the targets of an enzyme. And we now know that hypophosphatase is HPP is caused by low levels of the enzyme alkaline phosphatase. So we're missing something that's going to break down other chemicals. And the concept of enzymes might be most familiar to you if you're doing the wash. Here is a detergent, and some of these detergents contain enzymes in the powder that you put into the wash water. And here is an enzyme, and this particular enzyme has the job of breaking down fat stains, blood stains, grease or grit, because it acts by binding to the blood stain or the grease or the grit, and then somehow breaks the bond or breaks up the substrate, releases it, so it's smaller now, it goes into the wash water and rinse. But this is a nice example of one or a combination of different enzymes and how they work, let's say in the wash water, but in all of us we have hundreds and hundreds of enzymes within ourselves that are also regulating chemical levels. So yes, an enzyme is a protein that breaks down these substrates, and hypophosphatase is specifically due to deficiency of the enzyme alkaline phosphatase. Alkaline phosphatase assays, going all the way back to Robeson's day, could be adapted to looking at blood samples. You would get a tube of blood, you would spin it, there would be serum or plasma, you could take that off, you could put it into a so-called buffer, and using substances that turn color when they're broken down, you could measure the amount of color that would be formed, and if you use specific substrates for alkaline phosphatase, the more the activity, the more the alkaline phosphatase, the more the color was released. So now you could measure it not only in his laboratory, but you can measure it in clinical laboratories using blood in case it had some clinical usefulness. Of interest, Robeson found that this enzyme assay worked best at so-called alkaline pH in his laboratory. So what is alkaline? You know that there are acidic things, acid, there are alkaline things, alkalinity, and here pure water has a pH of 7, that's neutral, but you can become more acid as you go to saliva, to black coffee, bitter tasting tomatoes, vinegar, stomach acid or lemon juice, and then even hydrochloric acid. This is the acid side of pH. And then you could go in the opposite direction. You could go alkaline. Seawater has a slightly higher pH, baking soda even more, household ammonia, very high pH, bleach, oven cleaner, and then lye, or sodium hydroxide, has a very high alkaline pH. So when Robeson was testing those color-generating compound substrates, he found that this reaction worked best in the test tube in his laboratory when he was working at alkaline pHs in the buffer, and that's where the word alkaline phosphatase comes from. Phosphatase just means it breaks off phosphate, and alkaline says it does it at alkaline pH. So why do we assay blood alkaline phosphatase levels? Actually, we're not measuring the protein, but we're measuring how well the alkaline phosphatase we would say hydrolyzes or takes off the phosphate group and then becomes colorful in the test tube. That's a measure of its activity. Well, chemists ever since about the 1930s have recognized that doing this assay in a clinical laboratory in a hospital has importance. Why? Because high alkaline phosphatase levels in blood warn physicians about the possible presence of bone or liver or biliary disease. Biliary disease would be something wrong, let's say, with the gallbladder. So that's why when you go to the doctor and you get a comprehensive panel drawn, there's always alkaline phosphatase in it. It may be the most frequently assayed enzyme ever, and the docs are looking for high levels to make sure or looking at high levels, hopefully finding normal levels to make sure that you don't have problems here. But until recently, maybe it's because of the increasing interest in hypophosphatasia, until recently, physicians in laboratories really didn't pay much attention to low alkaline phosphatase levels. They weren't indicating this. We knew of some associated disorders, I'll show you them, but a low alkaline phosphatase wasn't nearly as startling to clinicians as a high level. And over the years, there were causes of low alkaline phosphatase levels identified in serum. And this is called hypophospha, and it's almost hypophosphatasia, but it's hypophosphatasemia. All this means is that there are low levels of phosphatase in the blood. So that not everything that causes low serum alkaline phosphatase is hypophosphatasia. That's certainly one of them. But people have studied families, and there would be some low alkaline phosphatases going from generation to generation or in siblings, and they would say maybe this is familial, benign, not associated with disease. Or low levels could occur with profound low blood counts, anemia, scurvy, severe forms at birth of osteogenesis imperfecta, multiple myeloma, a variety of other diseases, usually very severe conditions that physicians should recognize pretty easily. These are associated with low levels of serum alkaline phosphatase. So not everything that's a low alkaline phosphatase is necessarily hypophosphatasia. Back to Robeson, he continues his work from 1923 to 1932. Then he goes to New York University and he delivers an important speech summarizing the significance of phosphate when it combines with other chemicals, its role in metabolism. And in that publication, he makes a very important point toward the end, and he says that, yes, back in 1923, we thought that releasing phosphate from sugars might be providing some of the phosphate necessarily to form bone, but Robeson thought there's actually a second mechanism. Something else is controlling the deposition of bone salt. He didn't know what it was, but this is going to be a major factor in what causes the problems in hypophosphatasia. Robeson knew that the alkaline pH was not physiologic. None of us ever has a pH that is so high to resemble what was in his test tube. He never used the expression alkaline phosphatase, but instead, quite rightfully, said, we'll call this bone phosphatase. It was the chemists later on in the clinical laboratories who were using the alkaline pH that then started alkaline phosphatase, and that has stuck, and I think it'll be with us forever. We're always going to be talking about alkaline phosphatase. So the chemists keep working, and what do they find? In human beings, they find that there are three forms of alkaline phosphatase. I have to back up. I don't think this is working quite properly. There are three forms of alkaline phosphatase that are specific to tissues. One is found only in the placenta, one in the intestine, one in the ovaries or testes. These are called tissue-specific because they're only found here, 3 different alkaline phosphatases. But the 4th one in humans is ubiquitous. It's everywhere. And this is the one that you find in bone and in liver. And therefore, we call this tissue non-specific alkaline phosphatase. It's everywhere. And I like to call it TNS-ALP. That's the abbreviation for tissue non-specific alkaline phosphatase. And in 1979, this book was published. It talked about how you can measure it, what substrates to use, what animals have it, how you could separate the one in placenta from the intestine, and many, many things like that. But this 1,000-page book contained only 30 pages, 3%, that speculated back in 1979 as to what alkaline phosphatase really does. So back then we 4 types of alkaline phosphatase were really doing. But there was progress. Back in the 1950s, chemists had identified this, phosphoethanolamine, PEA, in the urine of patients with hypophosphatasia. In the 1980s, we found that the most prevalent form of vitamin B6 in the circulation, pyridoxal phosphate, or PLP, was elevated in hypophosphatasia. And importantly, chemists had identified in the 1960s that inorganic pyrophosphate, pyro means heat. If you heat phosphate, and you will combine 2 phosphate molecules to form inorganic pyrophosphate, PPI. This was present in high levels in the blood and urine of individuals with hypophosphatasia. And when you look at these molecules, and that's not allowing me to do that, here's the phosphate, here's the phosphate, here are 2 phosphates. And what alkaline phosphatase is doing is it's clipping off these phosphates. So these are the natural substrates being broken down by TNS-ALP. Where does pyrophosphate come from? Well, this cartoon, if it was working, would show you that ATP, a high-energy phosphate compound, is broken down by another enzyme, PC1, on the surface of cells, and the pyrophosphate comes out and goes into a pool. There's also a channel that kind of spits out pyrophosphate from inside cells, putting it into a pool. And then it's the job of tissue non-specific alkaline phosphatase to break down the pyrophosphate, 2 phosphates, into single phosphates, providing it for skeletal mineralization, formation of bone crystals, etc. So if you're missing in hypophosphatasia an active alkaline phosphatase, this is going to build up. Will that be a problem, having too much pyrophosphate? Yes, that's going to be the major biochemical problem in hypophosphatasia, or HPP. I'm trying to change slides, bear with me. Yeah, I consider inorganic pyrophosphate the villain, the chemical that builds up in hypophosphatasia, and we need to get it down into normal levels to treat the disease. So the villain in hypophosphatasia, I consider the buildup in the body of inorganic pyrophosphate, PPI. Why? Why is this a villain? Because PPI is a potent inhibitor. It blocks minerals like calcium and phosphorus from going into bones and teeth. So if the PPI levels are high because the alkaline phosphatase activity is low, you're not going to mineralize your skeleton or teeth properly. And what happens is, if you look with an electron microscope at the very first beginnings of mineralization in a healthy person, you have these little tiny matrix vesicles, and inside form hydroxyapatite crystals from calcium and phosphorus. And then these crystals grow, getting ready to be put into the fiber, the collagen. They grow until they rupture this vesicle, and then they should grow some more. There should be more calcium and phosphorus, bigger crystal into the bones, healthy bones. But in hypophosphatasia, the inorganic pyrophosphate is sitting there in the bloodstream and outside the cells, and it blocks these crystals from growing. So this is the problem, too much inorganic pyrophosphate sitting there keeping the skeleton and teeth from mineralizing. And how does this translate clinically? It translates to rickets. Instead of a nice, beautiful conversion of cartilage to bone. In children, there are these tongues of radiolucency. There could be widening of growth plates. So you could see it on an X-ray. Soft bones, that's where the foundation name comes from and what it tries to capture. Soft bones, rickets, osteomalacia when it occurs in an adult. And then in a child, the teeth are especially sensitive to the low alkaline phosphatase and the elevated pyrophosphate. And instead of the baby teeth being shed normally where the bone-chewing cells or tooth-chewing cells cut off the root and the child takes the tooth out at age 6, what's happening in hypophosphatasia, the patients get scared or the parents because the whole tooth slides out bloodlessly, painlessly, root intact. Why? Because there's no mineral on the surface of the tooth root and out it comes. So this is a very sensitive area of the teeth, the dentition of children with hypophosphatasia. And there is a form, odontohypophosphatasia. It's probably the most common form of hypophosphatasia, where only the teeth, but not the bones, are affected. So why are blood and tissue alkaline phosphatase levels low in hypophosphatasia? So what's gone wrong? Why is the alkaline phosphatase not working? Well, from our mother and from our father, we get a copy of the gene that makes tissue nonspecific alkaline phosphatase. And if we're inheriting from a mom or dad one defective copy, or perhaps clinically worse, 2 defective copies, one from mom and dad, of the gene that encodes, makes tissue nonspecific alkaline phosphatase, then it's not going to work. It's defective. The pyrophosphate levels will build up. And that is the cause of hypophosphatasia, which is a heritable, a genetically based disorder. So in your DNA is the cookbook sequence of nucleotides that are encoding many, many things, perhaps 20,000 genes. The gene for tissue nonspecific alkaline phosphatase is only one of them. But the coding should be perfect in order for the DNA to talk about how an amino acid chain that is the alkaline phosphatase should be put together and folded to make a nice happy alkaline phosphatase out here. And if you have a mutation, a defect of the gene that's making alkaline phosphatase, then this sequence is imperfect, the folding is improper, and then the alkaline phosphatase is not going to work. And there are many ways that the alkaline phosphatase molecule can be adversely affected. This is what it looks like under sort of an electron microscope crystallography. It almost looks like a bat with head and wings. But 2 molecules of alkaline phosphatase come together. Each one has a site where it can remove phosphate from these various compounds, break up pyrophosphate. But this has to be just perfect in terms of its amino acid sequence. The two dimers have to come together perfectly for all of this to work. It has to hook to the surface of cells. So anything that's disrupting the homodimer, the alkaline phosphatase, the mature enzyme, can lead to deficient activity. And why? How can we explain the remarkably broad-ranging severity of hypophosphatasia? Well, we now know from study of hypophosphatasia patients throughout the world that there are 300 or more specific mutations in the alkaline phosphatase gene that will lead to a defective monomer of the alkaline phosphatase enzyme. And you can inherit one defective copy of the gene and the alkaline phosphatase from mom or dad, and that's more likely to give you mild disease. or you can get a defective copy of the gene from mom and dad. So now you've got no alkaline phosphatase that's working perfectly. And it's this many, many different mutations and inheriting one at a time or two at a time that account for the broad-ranging severity of hypophosphatasia. So can you see this in how we measure alkaline phosphatase in the clinical laboratory? Here's a normal distribution curve. And if we drew blood from 1,000 people, healthy people, not everybody has a perfectly average alkaline phosphatase. They're all healthy, but some have high levels, some have lowish levels compared to the average, and 68% are going to be around here, but 95% are going to be here and here. So this is how statisticians define a normal range, but if you're over here, does that mean, and it's low, does that mean you have hypophosphatasia? No, not necessarily. This is actually just a little bit lower than the normal range, but 2% of people walking around are going to be here and not necessarily with disease, and 2% will have a slightly elevated level compared to this normal range, but that doesn't mean they have bone, liver disease. So in the chemistry laboratory, defining a normal range is very important because, again, if it's liver disease or bone disease and it's a high level, it's going to be somewhere out here or here. And if it's hypophosphatasia, I'll show you it's not going to necessarily be there, but it's going to be down here in general. So years ago, we got blood from patients with different forms of hypophosphatasia. This is a special scale, and these are healthy kids. And you see 100 to about 350 is normal, 100 to 350. So what does perinatal, the most terrible severe hypophosphatasia, show you? A value of 4 or 12, but not 100 to 350. Infantile hypophosphatasia might be a 10 or a 20, but certainly much lower than healthy kids. And the childhood form, severe forms within the childhood or mild forms, they're still low. They could be distinctly low. Here's a 40. That's much lower than 100 to 350. And in general, we think of any patient with hypophosphatasia should have a low serum alkaline phosphatase activity. Here are the adults. Here's healthy adults. And when you look at adults who have just tooth loss, odontohypophosphatasia, or have some fracturing, they're clearly lower than the healthy peers. So these are values that tend to reflect the clinical severity of hypophosphatasia, the classification or nosology. But all of this changes over time, and this is very important for all physicians to realize. Adults have this as their reference range for alkaline phosphatase. Different assays could be used, so you have to know what the normal range is for a particular laboratory, but in general, normal for an adult might be 30 to 120, and that's pretty steady during adult life. But when you get down to teenagers, they're growing rapidly, they still have open growth plates, and their normal range for alkaline phosphatase is much higher than adults. Young kids, say age 5 or 6, they're clearly elevated compared to adults. They're healthy. And babies, newborns, have very high levels as well. So anybody who's looking at an alkaline phosphatase and interpreting it must know that this pattern exists in good health, and to know that children are usually much higher than adults. And this normal range is key. Laboratories need to report it. Not all do. And you can get into trouble where if you have a child with hypophosphatasia and the value's over here, and the laboratory is only reporting out the normal range for adults, this gets reported out as normal, but it's much too low compared to children. So you can end up with a delayed diagnosis or a misdiagnosis unless the laboratory is really giving you almost year-by-year set of normal ranges. And there's been good progress of that. Most laboratories are now doing this. And here's a reference range. And look, it's broken down to 4-year-olds, 5-year-old boys. And here's a normal range. But look at how different a 4-year-old is compared to somebody 19 years old. And the girls are a little bit different from boys over time. So luckily now, using international units where the same assay is used, we have nice normal ranges to use in diagnosing high or low alkaline phosphatase levels. So in the last few slides, I could say that since 1979, nice progress in understanding mammalian, including human alkaline phosphatases. This was published by Jose Luis Millan from Sanford Burnham Institute against San Diego in 2006. You know that alkaline phosphatase has been used or been approached to treat hypophosphatasia over the years. In the 1980s, we gave plasma that was rich in alkaline phosphatase activity, infused it into babies with very severe hypophosphatasia, fixed the bloodstream but not the bones. We tried to give alkaline phosphatase-rich cells by marrow cell transplantation. We tried to increase the levels being made by the bone-forming cells by using off-label medicine for osteoporosis, teriparatide, with a little bit of success, but we realized all along that the goal was to increase alkaline phosphatase levels in the skeleton itself. So along came ENB 0040. Eventually this would be named asfatase alpha. And this is the treatment that you're probably hearing most about. This recombinant molecule, now that you could do things with DNA, would combine 2 alkaline phosphatases, and down here, a way of making the bone crystals take it up so that it's right in the skeleton or in the teeth where it needs to work. So this is recent progress in understanding where and how alkaline phosphatase is working for mineralization of the skeleton and teeth. You're all interested, I believe, in hypophosphatasia and also alkaline phosphatase, and I could advertise that it also in this month's issue of Nature Reviews Endocrinology, I provide for physicians and maybe scientists an overview of hypophosphatasia and alkaline phosphatase, the cause of the disease, how we classify it, the pyrophosphate as the villain that we need to lower the levels of, how we diagnose it, and how we treat it. And if you wish, here's some contact information. We're happy to see here at Shriners Hospital in St. Louis children with all forms of hypophosphatasia. We don't have an intensive care unit, so the perinatal and the very sick infantile patients with hypophosphatasia tend to be managed more locally, but we see the odonto and mild and severe childhood hypophosphatasia patients and those who have survived the infantile form. Feel free to contact me, and I give credit to my home university and Shriners Hospital here in St. Louis. We'll end here and be happy to answer questions.
Perfect. Thank you, Dr. Whyte. So no questions have come up yet, but I'd like to open up to all the attendees listening today. If you guys have any questions for Dr. Whyte that he can answer, please feel free to utilize the Q&A portion at the bottom of your right-hand corner. I
see one about somebody asking to be reconnected. I don't know if I can kind of scroll to look for other questions.
Oh.
Here's one. As I use this.
Would you like me to read them, Dr. Whyte?
Yeah, I think that would be easiest.
Sure. A question came in, how else is the body affected by lack of ALP besides bone and teeth issues?
In the very most severe forms, and this would be perinatal and infantile hypophosphatasia, there could be such a profound deficiency of alkaline phosphatase activity that the metabolism of vitamin B6 is disturbed in such a way that none of the buildup of the pyridoxal phosphate in the circulation, none of it is broken down sufficiently to make pyridoxal. That's the form of vitamin B6 that can get into cells. So in a sense, there's a tremendous extracellular buildup of PLP, but not enough for cells. And that could lead to a deficiency of neurotransmitter synthesis in the brain, so that very severely affected infants and newborns can have vitamin B6 dependent seizures. That's a hallmark of the biochemical disturbance and has been a marker for lethality. Before the availability of enzyme replacement therapy, all 18 patients who manifested such seizures went on to die from hypophosphatasia. A number of them who have had seizures are on clinical trials or being switched to the commercial enzyme Strensiq and have survived years without the lethality of the vitamin B6 dependent seizures. There's also, depending on the severity of hypophosphatasia, a muscle weakness. And I think of it as likely also a result of the excess pyrophosphate levels outside the cells. The reason I say that is that there are similarities with some of the drugs that we use for osteoporosis to pyrophosphate, and if you overdo it with those drugs, the weakness, the rickets and osteomalacia, and an elevation in blood phosphate are characteristic of that excessive exposure to those agents mimicking hypophosphatasia. I think last week at the Endocrine Society meeting in Boston, Alexian investigators may have presented, I wasn't there for the presentation, but they may have presented some evidence to support that notion.
Perfect. So the next question is, what is the percentage of patients with HPP?
The various forms or the prevalence in the world. I would say that the prevalence, and by prevalence we mean how many folks are born with the disease or have the disease, prevalence, incidence. It's clearly highest in Mennonites in Manitoba, in Winnipeg, Manitoba, because there's a founder mutation in that population, and it reappears and causes severe disease. There's also a so-called founder mutation in the Japanese, and when it reappears as recessive inheritance, inheriting one defective gene copy from mom and dad, it also causes severe disease. But in many other places where you're encountering either single mutations or combinations of different tissue non-specific mutations, and that explains the great broad-ranging severity. The prevalence of hypophosphatasia severe in the Mennonites was estimated to be about 1 in every 2,500 newborns. In Toronto, it was thought to be less, but about 1 per 100,000. In Europe, it was approximated to be about 1 per 300,000. But in Europe, the estimation for moderately severe disease was that it was occurring in about, I think it was about 1 in 6,000 individuals. So it varies from place to place, and it varies in severity. I think worldwide, in our experience, my experience, the most prevalent, the most common form is odontohypophosphatasia. This is the mild one with tooth loss only, and it's because having one defective allele, one defective copy of the gene, is enough to give you tooth problems that could go from generation to generation, whereas the very severe disease, infantile or perinatal, is combining two. So that's a less likely occurrence, and that's why those forms are rarer than odontohypophosphatasia.
It appears currently more adults are being identified versus pediatric population. Is there a reason?
I think in part is the explanation for that is what I just said. It generally takes two defects coming together to cause severe perinatal or infantile or severe childhood hypophosphatasia, whereas a single defective tissue nonspecific alkaline phosphatase, it depends on the exact mutation, but that's a likely explanation for the more mild forms that can be transmitted from generation to generation. So here at Shriners, we might have a child who comes to us with odontohypophosphatasia, but we're always very interested to explore whether or not grandpa, grandma, or great-grandma, grandpa have any manifestations of the adult form of hypophosphatasia. So it's the common presence of one defective allele that seems to explain why the more mild forms are more common than the severe forms.
Great. What is the major difference between HPP and hypophosphatemia?
Oh, very different. It's medical language. Hypophosphatemia means low phosphate. So you've got low circulating levels of inorganic phosphate PI. And that can certainly give you rickets serostea malacia, XLH, tumor-induced rickets serostea malacia. Other disorders have as a major pathogenetic causal problem renal phosphate wasting, so that the inorganic phosphate, the PI levels, drop in blood. So now that you've got maybe a normal amount of calcium, but you don't have enough phosphate in the blood to mineralize your bones. So that's hypophosphatemia, low blood phosphate. The word for low blood alkaline phosphatase is similar. That's hypophosphatasemia, and that can be very confusing, and importantly confusing. So those words try to separate the two, but with hypophosphatasia, you get rickets serostea malacia, even though the bloodstream has normal amounts of calcium and phosphate, whereas in hypophosphatemia, the phosphate is too low, and that's how you can end up with rickets serostea malacia.
Right. It looks like there's a lot of online references that list average ALP levels by age, and they vary widely. Is there a preferred resource?
No, I think these laboratories are using standardized instruments, standardized assay approaches. So, but also with each establishment of an instrument and bringing on board an assay, they're referring back to literature of their own experience to create normal ranges. So it isn't terribly surprising that the normal ranges can differ from, let's say, commercial laboratory to another, or a hospital-based laboratory.
Are you treating patients with OI with Strensiq?
No, and I wouldn't see a rationale for it. In osteogenesis imperfecta, most patients have a defect in the collagen, the fiber part of bone, and that would not be fixed by alkaline phosphatase replacement.
What does high PLP do in adults? Does it affect the brain in the same way as infants?
No, it's not the high PLP. That seems to be an accumulation that doesn't have consequence. In fact, we sometimes give vitamin B6 to study patients or carriers of hypophosphatasia to see just how high the level goes. It doesn't seem to have any ill effect. The ill effect comes in the very severe patients where they just can't break down any of the PLP to PL. It's the PL that gets in that of what we would say hydrolysis of PLP to PL, then you could get seizures. So it's a deficiency of PL, not the buildup of PLP that we think is trouble. In adults with more mild hypophosphatasia than perinatal or infantile cases, we think of the PLP levels as distinctly elevated, but they don't go as high as in the very ill babies or newborns.
Perfect. We'll go ahead and do just one more question, and then I'm going to go ahead and copy all of these questions and pass them over to Dr. Whyte and Denise, and they will be able to contact everyone. So if we didn't get to your question, hopefully Dr. Whyte or Denise will be able to email you guys all personally and let you guys know the answers to many of the questions you have. Some are a little bit more general as well as some specific. So we will make sure we get all those questions to Dr. Whyte so he can answer that for everybody. So for the final question, what about for adults? Other than bone and teeth issues, how does lack of ALP affect the adult body?
Other than bones and teeth, well, with the dental manifestations of hypophosphatasia in adults, that is an area that needs study. We know that the children lose teeth because they don't have mineralized cementum, they slide out. But it seems clear that adults with hypophosphatasia can have dental issues. It may be somewhat akin to what happens in some baby teeth where maybe the enamel isn't quite as strong or the dentin doesn't form perfectly. I think there's need for a considerable amount of research on adult teeth in hypophosphatasia. Of course, there's need for research for the pediatric dentition as well. So there's that poorly understood problem. I see most of the complications of adult hypophosphatasia being attributable to the underlying osteomalacia or adult rickets. And then you could have fracturing, microfracturing, some deformity, achy bones. So those complications, I think, are directly attributable to the osteomalacia. It's understandable how with debility, from fracturing, from achiness, you could have muscle weakness, problems like that, whether or not the muscle weakness is akin to the muscle weakness that you see prevalent in children. That will need more study. And then in adults, it's very interesting that in contrast to children, the long-standing elevation in pyrophosphate outside of cells in the blood can lead to various types of arthropathy, arthritis. And adult patients with hypophosphatasia can have pyrophosphate arthropathy. They can have joint difficulties, bone destruction within joints. somehow attributable perhaps to the accumulation of crystals of inorganic pyrophosphate. Some of these patients have pseudo-gout. It resembles the gout of uric acid accumulation, but here the culprit once again is accumulation of pyrophosphate in joints, so pseudo-gout. And then there's a not terribly well understood problem of calcific periarthritis, where it's paradoxical that around shoulders or elbows or hips, there can be depositing of hydroxyapatite crystals, not where they should be, inside bone, but around joints. There's that, calcific periarthritis. The pyrophosphate can combine with calcium and calcified discs, vertebral discs, maybe predisposing to disc degeneration. And then also poorly understood, sometimes tendons and ligaments calcify when they shouldn't be. So here you have a problem where your bones aren't mineralizing properly, but ligaments and tendons, joint capsules are calcifying excessively. So those are complications that the adults see that the children don't.
Wonderful. Thank you, Dr. Whyte. Again, we are coming to the end of our webinar. So if you guys have any further questions, I will leave the webinar open for a couple more minutes. Please feel free to type in your questions, and I will be getting all of those questions to Denise and Dr. Whyte to be answered offline. So to close us up for today, I'm going to go ahead and pass this off to Denise.
Good afternoon, everybody. This is Denise Goodbar, and I'm the program manager for Softbones, for those of you who I haven't had the pleasure of speaking with. First of all, I wanted to give a big thank you to Dr. Whyte for providing us with such a worthwhile presentation. I know I learned a lot, and I'm sure you all would concur. I would also like to take the time to thank Global Genes for providing the technical support. We certainly couldn't have done that without you. As the Softbones Foundation and community, we're very lucky to be able to provide you with programming like this, and hope to continue to be able to provide you with worthwhile webinars like this going forward. As Ashley said, please feel free to email me any additional questions that you might have at denise@softbones.org, and I will be happy to coordinate with Dr. Whyte. So please reach out to me, not to Dr. Whyte directly, and I will be happy to compile the questions and answers. And we'll be sure to get you that information. Again, thank you so much again to all of you for participating, and look forward to future worthwhile events. Thank you.